Hi class, in this recording we're going to be focusing on the electron transport chain and how we use it for oxidative phosphorylation. Oxidative phosphorylation is a nice way of saying that we make ATP at the end. This is a name for how we make the ATP. So when we think of the electron transport chain, to bottom line, the ETC, the electron transport chain, its purpose is to make ATP and make lots of it. We want to pump out as much ATP as possible. So to recap, we started off with glucose entering the cell. That glucose enters this glycolysis and is broken down into pyruvic acids. Each of those pyruvic acids is sent to the mitochondrial matrix where they are processed in the citric acid cycle and have lots of high energy electrons removed. Those high energy electrons, as they are removed from the cell, pyruvic acids that have been entering into the citric acid cycle, are then sent to the electron transport chain. And it's at the electron transport chain that the high energy electrons are used to make the ATP. The electron transport chain takes place on the inner membrane of the mitochondria, known as the mitochondrial cristae. And the mitochondrial cristae has a series of proteins that are linked together. Protein 1, 2, 3, boom, 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 bada bing, bada boom. And these linked proteins will have the high energy electrons being transported between them. The high energy electron carriers are NADH and FADH2, and they carry, so this should not be positive right there. That's a typo on my part. And they carry the electrons to the electron transport chain. While those electrons are being dumped into the electron transport chain, the NAD and the FAD, or NADH and FADH2s are regenerated. That's a radish. That is so cool. Tim?
There's some radishes that are coming out of the ground.
That's awesome. You should make a French salad for lunch today.
Yeah.
And Tim, I'm in the middle of recording, so I gotta let you go. All right, so where were we, class? So the NADH and FADH2s are turned back into NAD positives and FAD positives, so that we're regenerating the electron carriers. You can think of these electron carriers as kind of like wheelbarrows. The empty wheelbarrow goes over to a glucose or the citric acid cycle, and then will pick up a high energy electron, and then carry that high energy electron over to the electron transport chain, dump it off, and then you have an empty wheelbarrow again that you carry back over to glycolysis or the citric acid cycle to get more high energy electrons. The energy in those high energy electrons is used to phosphorylate ADP. So to add a phosphate group, to phosphorylate is to attach a phosphate group. And if we attach a phosphate to adenosine diphosphate, we make it adenosine triphosphate. We go from 2 phosphates to 3 phosphates. So here's a series showing a figure that I appreciate that just shows how this process occurs. We have our NADH dropping off a high energy electron and being regenerated as NAD positive. FADH2 drops off its high energy electron, excuse me, and then becomes FAD positives. And then these high energy electrons are going to be shuttled from one protein to the next to the next. And as these electrons are being shuttled from protein to protein to protein, they're losing energy. Ultimately, they want to go to the lowest possible energy state. The lowest energy state is the most stable energy state. And that most stable energy state is when they bind to something that is highly electronegative or has a high propensity to attract electrons towards its nucleus. And in this context, we use oxygen. Oxygen is highly electronegative and has a strong attraction for electrons. And if we take half of an O2 molecule and combine it with 2 protons from the mitochondrial matrix, and then we combine it with 2 electrons from the electron transport chain, we make water. So we actually produce the water that our body needs. Not all of it. We do need to drink water. There are some organisms that get all of their water from the electron transport chain. We are not one of those organisms. We do need to drink water. But it's worth emphasizing that this water that's produced is a significant source of our body's water. It's also worth emphasizing that during this process, we don't always make water. Sometimes instead of making water, there's a little bit of a mistake and we make an H2O2, a hydrogen peroxide, or we make an O negative 2, a superoxide. So right here, this is where we produce a lot of the free radicals of our body. H2O2, hydrogen peroxide, is one of the most common free radicals in our bodies. Now, you probably remember from your high school classes the law of conservation of energy. Energy is not created or destroyed, it only changes form. So if we take high energy electrons and we lower their energy state, what do we do with the energy? How is the energy transformed? We take that energy and we turn it into a chemical concentration gradient. And as these electrons are bouncing around, we're going to take high energy, or we're going to take protons, or H+1 ions, from the mitochondrial matrix, and then pump them into the intermembrane space. And we build up a high concentration of protons in the intermembrane space. So we actually have an acidic pH in this intermembrane space. And these high concentrations of protons are then used to power something else, ATP synthase. So we have a concentration gradient. We have a high concentration, H for a high, in the intermembrane space, and then we have a low concentration of protons in the mitochondrial matrix. And chemicals tend to flow from an area of high concentration to an area of low concentration. And the chemicals will flow physically through a tube in a protein referred to as ATP synthase. As we look at this protein, it ends with ASE. That means it's an enzyme. What does this enzyme do? It synthesizes ATP. And while we're looking at ATP synthase, this ATP synthase, as it has protons moving through it, will physically spin. And the physical spinning of ATP synthase allows for it to grab an ADP and a phosphate group and physically smash them together to make ATP. This process at the end is called oxidative phosphorylation. And this occurs at the ATP synthase. There's little skull and crossbones right here. And that little skull and crossbones is significant because this terminal protein, this third protein in the electron transport chain, is oftentimes a target site for poison to bind. Just want to advance one slide quickly here. There we go, cyanide poisoning. And the reason I had that little skull and crossbones right there was because if somebody is poisoned with cyanide, that cyanide will bind to an iron in that third protein of the electron transport chain. And it prevents electrons from moving. And if the electrons stop moving, it's going to cause a traffic jam. And ultimately, we lose the ability to drop off electrons. And if we lose the ability to drop off electrons, we lose the ability to regenerate electron carriers. And if we lose the ability to regenerate electron carriers, so I'm back on this figure, if I can't drop off electrons right here because I've blocked it with cyanide, this protein becomes saturated, and then this second one, and then the first protein becomes saturated, and then we have nowhere for the NADH and FADH2s to drop off electrons. And then if we can't regenerate our electron carriers, the citric acid cycle shuts down entirely. And then we're left with only glycolysis and anaerobic respiration to produce ATP. So in essence, when somebody is poisoned with cyanide, it's like they are being asphyxiated. They lose the ability to use the oxygen they breathe in and breathe out. And consequently, they die from lack of ATP. As we work through this process, the electron transport chain proteins are going to be pumping protons into that inner membrane space. And as they move those protons into the inner membrane space, we have that high concentration of protons that will move through the ATP synthase. As we look at this process, for those of you interested in bookkeeping, for every 6 protons that move through the ATP synthase, we make 2.5 ATP, which is kind of goofy from a bookkeeping perspective. The reason we have this bookkeeping is if we drop off one electron from an NADH, and then we drop off that one electron from an NADH, will result in 6 protons in the inner membrane space. Another way of thinking of this is for every one NADH that we drop off, or electron that we drop off from an NADH, we make one water molecule. For every 2 electrons that NADH will drop off 2 electrons, and we use the 2 electrons to make up 1 water molecule and 2.5 ATPs. No, you do not need to know this bookkeeping for the exam. I find it interesting. Personal interest. That's all we have for this recording on the electron transport chain. If you have any questions or comments, please feel free to post them in the class discussion board or shoot me an email. And as always, class, happy studies.